Hello, good morning, everyone. It's good to be back this week. So we have a lot to do today. First, we have to take up the Lecture 8 study guide, which has a lot of problem solving involved in it. So that might take us some time. And then we will be taking up some of the questions from quiz number 3. I mean, the class average was pretty good, 75 or 76 or something like that. So it's significantly better than the previous one. But we'll take up some questions from there, and we will also talk about the lab. All right. Yeah, let's get started then. First thing is this is inheritance, which involves the concept of meiosis. So maybe let's take a moment and review about what meiosis actually is. Okay, so over here, meiosis. Okay, we start with a cell right here. Okay, and this is what we call a germ cell. Germ cell. A germ cell is a cell that will become a cell that will become a sperm or an egg. So in male, germ cells are found in the testicles, and in female, the germ cells are found in the ovaries. Okay, so testicles and ovaries, they are called gonads, right? Gonads. We'll say found in the gonads, which is basically reproductive organs. Now, for simplicity's sake, we are going to just be drawing 2 sets of chromosomes. Okay, so we will use red here. That is chromosome 1 from mom. Okay, and then you get a chromosome 2 from mom. And then we will have chromosome 1 from dad and chromosome 2 from dad. Okay, so this cell is a diploid cell, okay, which means it has 2 sets of chromosomes. One set of chromosome is called N. 2 sets of chromosome, we say 2N, diploid. Okay, and then what's going to happen is, before we do the meiosis process, let's talk about the relationship between some of these things. So these 2 pairs, right, this one here and this one here, they are the same length, same size. They contain the same set of genetic informations, but possibly with variations in it. Does anybody remember what do we call these chromosomes? The 2 chromosomes, one here. What is the relationship between them? How do we refer to them? That's right. You can title it homologous. Okay, homologous chromosomes. Okay, so same size, same length, okay, same order of genetic information. Okay, and again, oops, important to know that it is we inherit 1 from each parent. Okay, so 1 from mom, 1 from dad. So we would have a total of 22 pairs of homologous chromosome. Chromosome 1, chromosome 2, all the way to chromosome 22. If you are female, then your sex chromosomes are also homologous because it's XX, right? But for male, it's X and Y. So the sex chromosome would not be considered as homologous in male. All right. So in this case, we would have 4 chromosomes. 4 chromosomes. Right. And what's going to happen is we will have DNA replication, just like before. DNA replication. All right. So after DNA replication, we are going to have 2 chromosome 1 from mom now. Then they're of course stuck together at the centromere. I'm going to have 2 chromosome 2 from mother. And then same thing for the father chromosome, paternal chromosome. Okay. So there we go. This is the centromere. Centromere. Okay. I'm just going to try to label some of these things for you. Okay, so centromere is just the location where they're stuck together, right? Now, what about these now? Okay, what do we call them? They are stuck together and they are genetically identical copies of each other, right? So we call them sister chromatids. Okay, identical copies. Okay, so sister chromatids. Sisters stick together. That's how you can remember them. Now, something that's different in mitosis and meiosis is that after DNA replication, the homologous chromosome will find each other in the process of meiosis, okay? And they will form these set of 4 things that are close together, and we call them tetrad. Okay, so homologous chromosome pairs, which is a homologous chromosome, will find each other to form tetrad. Okay. Now, just for comparison's sake, you don't have to draw this. If this was mitosis, then you would have the chromosome here, right? And then they would just be randomly all over the place, right? They will not find each other. The homologous chromosome will not find each other. Okay? They would just be like, around on their own. Okay. Does everybody understand what tetrad is? And what we're doing here. Okay, thank you. So why do we have to form the tetrad? What's the point? So what happens is when the homologous chromosome find each other and that they are in close proximity like that, what they could do is they could engage in something called crossing over. Okay, so I'm just going to try to draw this. Give me a second. Okay, so in crossing over, some of the genetic material from the paternal chromosome will exchange with the maternal chromosome. Okay, so a bit of a blue will exchange with the red one like this. Okay, and then similarly, the red one will go on the blue one. Let me try to draw this as best as I could. There we go. Okay. So over here, right, we call this crossing over. Crossing over, which is exchange of genetic material between non-sister chromatids. There is no point in exchanging material between the sister chromatids because they're identical, right? So you will only be exchanging it with the non-sister chromatids. And also, it's important to know that you are only doing crossing over between the homologous chromosomes, right? Chromosome 1 will not be exchanging genetic material with chromosome 2. Okay, you must be exchanging genetic material with the chromosome of the same type. Okay, but between the non-sister chromatids. Now, in almost all the textbook, they always show it like this, right? With just a little bit of blue and just a little bit of red. It's important to understand it's not just the bottom part of the chromosome that gets exchanged, right? Like in reality, what's going to happen is more like this. And like you would have your your original blue chromosome, for example, and afterwards it looks more like this. Okay, it looks more like, you know, oh, sorry, looks more like this. Okay, like it's going to be like candy cane. All right, it's going to be blue with a bunch of red all over the place. Oops, sorry, I don't know why I did the first one. Should not have. Okay, so this is before crossing over, and then after crossing over, it's just going to be like all mixed up. Okay, with red all over the place. My point is just that it doesn't just occur at the tip. Okay, it occurs like along the entire length of the chromosome. And same thing for the red one, right? It's going to have blue all over it. Okay, and this process, this crossing over, the purpose of it is to basically create genetic variation. Okay, so creates genetic variation, which is why we do not look like, you know, our parents, like identical. We're not like 100% identical to our parents. We look different. Everybody looks different. Okay, and part of the reason is because of this crossing over. Okay, and then this thing is going to divide, right? Okay, you're going to divide twice, and in the end, you're going to end up having haploid chromosomes. Okay, so then you would have 2 rounds of division. Okay, resulting in 4 genetically different haploid cells. Okay, that's it. All right, so that's a little review to get you all warmed up. Let's go to the study guide right now. Identify the meiotic stage in each of the following diagram. Okay, first one here, okay, probably is the easiest to tell because you see 4 cells, right? Okay, these are, and they're going to probably separate later, okay, because we see 4 cells. Then we know we are in the telophase. Telophase number 2. Okay. This one is a little bit tricky. Anybody want to guess what that is? Or not guess, but like, you know, anybody knows what that is? All right. Now, this one, okay, so we have a metaphase 2. Okay, yeah, that's good try. This one, we actually have to divide the cell first. Okay, so there's 1 cell right there. 1 cell right here. And then we have another cell here, right? Okay, they're not really separated yet. They will eventually. And if you just focus on one of the cells, you can see the chromosomes are being pulled to the different parts, right? Different ends of the cell, right? So each of these are actually undergoing anaphase. And because we have 2 cells right now instead of 1, then we know this is anaphase number 2. Okay. Anaphase number 2. Okay, so over here, we don't see any division. This is a single cell yet, right? Still a single cell. And we see the chromosomes being pulled to the opposite end. So this would be anaphase. Anaphase number 1. Okay. 1 cell. So we know it. We're still talking about the first meiotic phase. And we can see the chromosomes, but they're not arranged in any particular order. They're not in the middle. They're not being pulled to the opposite side. So we can say with confidence that this is going to be prophase. Prophase number 1. Over here, we can see that the cell is, you know, separated like that. and the chromosomes are not really together yet. Okay, and they're divided into two cells here. So we can say this is telophase part 1. Although it's you can kind of argue that it's a little bit hard to distinguish between telophase 1 and say prophase number 2. They do look kind of similar. I would say that telophase 1, the chromosomes are not as concentrated, okay, as they should. So over here, 2 cells again, and they're in the middle. So this is metaphase, metaphase 2, metaphase 1, and over here this would be prophase 2. Okay, that's it. Next one, we have a chart here. It's really quite simple. And let's just do it in a generic sense, and hopefully it will make sense. We have starting with a diploid cell. Okay, remember only I should put a note here. Only diploid. Hold on. Not necessarily diploid. Only. Sorry, I'm just trying to think about how to phrase this. Only even set of chromosomes can undergo meiosis. Okay, so like if you have 2n, then you can divide and then it becomes n and n, right? If you have 4n, then you can divide and become 2n and 2n. Okay, some organisms have 4n's. Okay? Like bananas, I think it's like octuploid. Okay, eight n's or something. So if you take 8n and you divide, you get 4n's and 4n's, right? If you have odd number sets of chromosomes, you cannot do meiosis. Okay, if something has 3n, there is no meiosis. Right? You cannot do 1.5n and 1.5n. Right? That's not going to happen, right? Okay, so for organisms with like odd number sets of chromosomes, they would not be capable of doing sexual reproduction. Okay? They would have to do asexual reproduction themselves. Okay, so that's something to make a note of. So over here, we again let's do a generic case. We start with 2n, okay, and then after DNA replication, okay, rather than doing the n numbers, maybe let's just do the chromosomes. We have 46 chromosomes. After DNA replication, it's still 46, even though they are doubling the amount of genetic material, okay, because they're still stuck together. We still call it a chromosome, right? And then you would divide, so it's still 46 and 46. And then afterwards, you will have 23, 23, 23, and 23. Okay. So starting here, we have 2n, 2 sets of chromosomes. After you double, now you have 4 sets of chromosomes, right? Even some of them are stuck together. And then after first division, you will have 2n again, back to 2 sets. And finally, at the end, you will have just 1 set. Okay, so 1 set in the sperm, 1 set in the egg, and if they fertilize each other, then you would get back a diploid organism, right? So over here we have humans. Chromosome number, is it haploid or diploid, right? So this is gonna be diploid. 46 is diploid, 2 sets. Number of pairs of homologous chromosomes, that's gonna be 23. Okay. After meiosis 1, how many chromosomes do we have? Well, after meiosis number 1, we are going to have 46, right? Oh, sorry. So sorry. I made a mistake here. This is 23. This is 23. Because, sorry, I know I'm confusing you guys. Okay, let me try that one more time. Okay. This is the chromosome, right? Okay, and it's going to be a tetrad. Okay, it's going to be a tetrad like this. Okay, and then after they separate, after it separates in meiosis one, okay, what's going to happen is you're going to have half of the tetrad here and half of the tetrad here, right? Okay, so that's still considered as one chromosome. Okay, but now they're separated. Okay, so that's why it's 23 and 23. You have the genetic amount, right? Okay, so over here, 2 chromosomes. After you separate like this, then it's 1 chromosome and 1 chromosome. Okay, so after the first division, it's already 23, but you still have like two sets over here, right? Okay, 1 set and 2 sets. So that is going to be 23. And the number of chromosomes after meiosis 2 is also going to be 23. Okay. So the idea is you start with 46, but then after each division, it's going to be half of the original. And so if this is 18, right? That means I started with 36, and then you will have 18 pairs, and then 18, and then no more homologous chromosome. Okay, so it's just the first number is always going to be double. Okay, and after each division, it's going to be half of the original. Okay. Sorry about the confusion there. I hope I'm making sense. Okay, let's talk about the next page, which is the table here. Comparing mitosis and meiosis. So first, the number of divisions in mitosis. There's only 1 division in mitosis. But for meiosis, it's going to be 2 divisions. Mitosis, we have 2 daughter cells. And for meiosis, it's a little bit different for male and female. In male, we're going to have four sperms from 1 germ cell. But for female, it's just going to be 1 egg. 1 egg plus 3 polar bodies. Okay, so just to refresh your memory, we have the germ cell. And then what happens is the first division, you would have uneven division of the cytoplasm. So 1 cell would get more cytoplasm than the other. Okay, and then same thing for the second division. Okay, uneven cytoplasmic division. This one might divide again. You have 2 smaller ones. So these are called polar bodies. Okay, you want the egg to have as much cytoplasm as possible. That's why you want to make sure you have one that's really, really big compared to the other ones. Okay, so in meiosis, you get 1 egg and 3 polar bodies. Next one, mitosis. Daughter cells are going to be diploid, 2 sets of chromosome. For meiosis, it is going to be haploid. Number of chromosomes in daughter cells for, if we're talking about, it is talking about humans over here, humans. 46 and 23. Prophase versus prophase 1. So in prophase, what happens is, well, basically nothing much happens. Homologous chromosomes do not pair up. Okay. Prophase 1, what happens is homologous chromosomes form tetrads. Okay. Crossing over occurs to create genetic variation. Okay. In metaphase 1, the chromosomes, they line up in the middle, right? Like this, right? Okay. So sister, well, we'll just say chromosomes line up at the center. Whereas for metaphase 1, the tetrad line up at the center. Center of the cell, right? Of course. Okay, next one. Genetic relationship of daughter cells to parent cells are identical. Whereas over here, it is variant, not the same. Type of cells involved, basically anything that's not germ cells. So somatic cells. Somatic cells means bodily cells. Of course, there are exceptions, but like brain cells, they don't divide, right? Okay, somatic cells. Germ cells. Function of mitosis is for growth and development. Repairing. Damaged tissues. For example, you have a cut, you need to make some cells. And what's the function of meiosis? Sexual reproduction. All right. Are we okay? Any question before we move on? Okay. Let's try this one. Oh, I forgot to launch. Nobody told me. Sorry. All right, let's end the poll. Produced by the process of mitosis. That's not true. It's supposed to be meiosis. Okay. Gametes is sperm and eggs, right? So contains 23 chromosomes. Produced in the testis and oviduct. It's not oviduct, it's ovary. Okay, for those of you who don't know, this is the ovary. That's where the eggs are made. And then the eggs are transported through the oviduct, okay, or the fallopian tube, right, all the way to the uterus. Okay, so this is the uterus where the baby is going to grow. Okay, so fertilization happens in the oviduct or fallopian tube. Right? So the sperm has to swim all the way up and meet the egg here. Okay, and then if fertilization occurs, it implants in the uterus. Okay, so this is not true. Contains haploid number of chromosomes. That is correct. Okay, that's the correct answer. Primary spermatocytes and primary oocyte. That's the starting germ cell. That's the name of that. Okay, so let's try this one. Ovum is just a fancy word for egg, okay? Okay, anyone else? Okay. Yeah, most of you got it right. I showed you earlier that the polar body can sometimes divide as well. Right? Let's take a look at over here. Right? So if this polar body also divides, then you end up having 1, 2, 3 polar bodies. Okay. Okay, last one. Actually, no, we got a couple more. But let's pick up the pace, okay, guys? Because we do have a lot to do. Oh, I just noticed there are 2 of the same answer here. Let's just ignore this one. Okay. It is going to be halved during the first meiotic division. Okay, so remember, right? 46, and then after DNA replication, it's still going to be 46, and then it's going to be 23, and then the split again is 23, 23. Okay, I know I confused you earlier, but this is the correct answer, okay? So copy this if you need to. This is meiosis 1, and then this is meiosis 2. Okay. So sorry again about the confusion earlier, but please write this down. Okay, take a picture, take a screenshot. Okay. Okay, let's try the next one. Make this a little bit smaller. It's kind of in the way. Or I can move this down. Okay, let's try that. Sorry. Oh, now I really messed it up. Let me just erase this now. Okay, let's try this. Okay, 2n equals to 16, which means n equals to 8, right? Okay, so one set equals to 8 chromosomes. So let's see what this says here. A lot of people chose A. Well, not a lot, only 7 of you answered, but like half of you chose A and the other half chose D. The species has 16 set of chromosomes. Okay, so now I just showed you, right? One set is 8, right? A set is the n number. A set is referring to the n numbers, okay? But if n is 8, right, then you would have 8 pairs of homologous chromosomes, which is why this is correct. Okay, that's the correct answer. All right, that's it for the meiosis. Let's move on to the Mendel stuff. First one, state the law of independent assortment. Okay, so Mendel, right, the so-called father of genetics, he has 2 laws. The first law is the law of independent assortment. Okay, basically, what it says is this: the alleles, okay, and alleles, if you're not familiar, alleles are basically 2 or like different versions of a gene, right? Okay, so for example, we can have a gene for eye color. So one version would be blue eyes, another version would be brown eyes. So these are 2 different alleles for the eye color. Okay, you can have a gene for blood type. Right? We can have allele for type A blood and allele for type B blood and allele for type O blood, so on and so forth. Right? So alleles are different versions of the gene. So the alleles of 2 or more genes get sorted into gametes independently of one another. Okay. But what does that actually mean? Okay. So for example, let's say I have a maternal chromosome here. You know what? Let's stick with our original red and blue scheme so we don't get confused. So let's say we have chromosome 1 from mom and chromosome 1 from dad. Okay, so let's say we are tracking 3 genes here. One gene is at the top, one gene is near the middle, the other is at the end. Okay, so like for the mother, there might be a capital A, small b, and a small c. And then for the father, it might be small a, small b, capital C, for example. Okay, so for this individual, okay, let's say this is a male. Okay, so when this guy undergoes meiosis and create the sperm, right? Because of crossing over during meiosis 1, these genetic material might get swapped, right? Okay, so in the end, what's going to be the possible combinations? Well, there could be a big A, small b, small c. You know what? I'm just not going to draw the sperm because it's taking up too much space. So we can have like big A, small b, small c. Okay, we can have big A, small b, big C. We can have small a, small b, small c, small a, small b, big C, right? So whether you get a big A or a small a for this gene will not affect whether you get a big C or a small c. That's basically what this law is saying, right? Okay. Now, the B doesn't matter because it's the same, right? So, there's no difference there. But let's just say I change this to a big B, right? Okay. So basically, the law of independent assortment states that which allele you inherit for one gene will not affect which allele you'll be inheriting for the next gene. Okay. So if you have 3 genes, if you look at 3 genes and all of them are different, then there are a total of possible 8 outcomes that you can have. And so I listed 4 here, right? And then I can list 4 more. Okay, so there are 8 possible combinations. Right? It's like saying this, right? If I have 2 separate coins, I flip one coin, whether I get a heads or tails on this coin will not affect whether I get head or tail on the other coin, right? Do you understand? That's basically what the law of independent assortment is saying. But rather than saying it in terms of the coin, we are talking about the individual genes. Okay, so there are 8 possible outcomes, right? 2 possible outcomes for the first gene, 2 possible outcomes for the second gene, 2 possible outcomes for the last gene. So that's going to be 8 possible different sperm, okay, or eggs if you're talking about females. Okay, so that basically allows genetic variations, right? Okay, next one, the law of segregation. The law of segregation states that during metaphase, basically, okay, only one of the 2 alleles in a diploid organism will be passed on to each gamete. Okay, so even though we each have 2 sets for each gene, we will only be passing on one of them. We know that already. Okay. All right, next question is a little bit tough, but let's try to do this. Okay. Examine each of the pedigree. Pedigree is these family trees, right? Okay. And determine whether the inheritance pattern is autosomal dominant, right, or autosomal recessive. Okay. And just to make sure everybody knows what dominant and recessive means. Dominant means you only need 1 copy for the trait to show up. Recessive means you need 2 copies, right? Okay. So let's take a look at this here. If it's not shaded in, then that means the person does not have the trait. If it's shaded in, that means the person has the trait. So over here, how do you know whether it's dominant or recessive? Well, one of the telltale sign for autosomal recessive is that you would have someone who possess the trait, but the parents do not have the trait. Okay, so if you see someone showing the trait, but the parents don't, then that's a key indicator that it is going to be autosomal recessive. Okay, if we're just trying to decide between recessive and dominance, right? So let me write that out for you over here. Okay, so autosomal recessive. So 2 parents without the tray have a child. We don't know if it's a kid, have a child with the tray. Okay. So how is that possible? Well, the parents could be like big A, small a, big A, small a, right? Okay, so if it's a recessive tray, the parents will not be exhibiting that tray. Okay. So because they have one of the capital allele, right? But then when you do the Punnett square, right, big A, small a, big A, small a, right? Then you can see that there is a 25% chance that their child is going to have the recessive trait. Okay, so an example of this would be, let's say both parents have brown eyes. Okay, brown eye is dominant over blue eyes. Right? Darker pigmentation is always dominant over the lighter one. So if you have if both parents have big A, small a, right, they would both have brown eyes, but then there is 25% chance that the child is going to have blue eyes. Okay. So over here, right, we can see that this is the only person who has the tray. So let's just say small a, small a. Then the parents here, they are going to definitely be big A, small a. Okay. What about the siblings? Well, we don't really know. Okay, they could be big A something, but there's no way for us to tell whether they are homozygous, right, big A, or heterozygous, which is big A, small a. Okay, so this is going to be recessive. Any question? Okay. What about the next one over here? Is it dominant? Is it recessive? Well, let's find out. Okay. You can start by assuming it is recessive. Okay, and then we'll see if it makes sense or not. So if I assume it's recessive, okay, assume recessive. Let's see what happens. So if this is recessive, then what is the genetic makeup of the generation 1 parents? Can someone tell me? If this is a recessive trait, what would be the genetic makeup of the generation 1 people? Yeah, they would be small a, right? Because that's the only way for the trait to show up if it is recessive. Okay? I need to know if all of you understand what I just said. Because you ask me a question, ask me to clarify it if you don't understand. Because if you don't understand this, everything else I'm about to say will not make sense either. Okay? So my question is, do you all understand why, if we assume it is recessive, then these people must be small a, small a? That's also small a, small a, small a, small a, small a, small a. Basically, if you're recessive, if a trait is recessive, in order for you to show that trait, to exhibit it, right, to express that trait, you must be homozygous. Okay. Now, so if this is recessive and they are both small a, then that means all of their kids, all of their children should have also expressed the trait, right? Yeah, because that's the only thing that they could pass on. 100% of their kids must also be small a, right? But as you can see, none of them actually have the trait. So that means our original assumption is incorrect. It cannot be recessive. Okay, so I'm gonna make a note here, right? Cannot be recessive because none of the generation 2 individuals have the tray, even though the parents have them. So for that reason, I know this is dominant. Okay, so I'm just going to fill it in for you. If it's dominant tray, then this is big A for sure. Okay, but is it big A, or is it big A, small a? Now, if they are both big A, then again, all of the kids would have that, right? Because they would only be able to pass on the big A, right? Okay. Then you might be thinking, okay, maybe one of them is big A, the other is big A, small a. That's also not possible, because if one of the parents is big A, then again, all the kids would have to have that tray already, right? Okay. But none of them do. So that's why I know it's big A, small a. Okay, so all of these is small a, small a, small a, small a, small a, small a. None of them has it. Okay. Over here we have a guy who has the tray. Okay, so it's gonna be big A something, but is it big A, small a, or big A, big A? Well, we're gonna have to look at their kids, right? So these are the children of this couple, right? Okay. And you can see there is one person who does not have the tray. Okay, so that means this is going to be small a, small a. And one of the small a came from the mother, which means the other small a must have came from the father. So for that reason, we know it is going to be like that. Okay, and these guys are all like big A, small a, big A, small a. Okay. Last one. Okay. Long story short, you should be able to tell right away that this is going to be, you know what? I'm going to do a poll just to see. I want everybody to answer, please. If you think is dominant, please choose A. If you think is recessive, please choose B. And if you're not sure, that's okay as well. Let me know that you're not sure by choosing C. Okay. 14 out of 7 of you answer, which is good. The correct answer is recessive. Well, how do you know? How do you know? Because of what I said here. If you see two parents without the tray means not shaded in, right? And they have a child, just one. If they have one child that has the tray, then that's a dead giveaway that it is going to be recessive. So over here, right? That is the giveaway, okay, of why this is recessive. It's not possible for this to occur if it's dominant. Okay, it's not possible for this to occur if it's dominant. All right, that's it. You know, today I think we're just gonna soldier on and keep pushing forward. Let's skip the break. It's just because we have so much to do. I don't want to, you know, not be able to finish everything. If you do need a break, help yourself, and you can just watch whatever you missed, you know, in the recordings. All right, I am going to take up some of these questions, but it's better if you let me know which one you want me to take up. Okay, I don't know if you guys had a chance to work through these things, but you know, for those of you who did, do you have any requests for the first part here? Is there a particular question that you want me to take up? No? Okay, I'll just take up some random ones then, okay, that I think is particularly good. So let's do number 4. Number 4. Oh, number 2? Okay, sure, we'll do that too. Let's do number 2 first, and then we'll do number 4. Okay, I'm gonna have to start a new page here. Okay, black coat color in guinea pig is dominant over white coat. Okay, so I'm gonna say black coat. Black coat is big B. We use capital letters for dominant, and white coat is small letter. Okay. Show the results of phenotype. Phenotype is what they look like. Genotype is what their genetic makeup is of the following cross. So let's do A. A hybrid black pig. Okay, what is a hybrid black pig? Hybrid means different allele. Okay, so for it to be a black pig, it can either be big B, or big B, small b. If it's big B, it's black and it's pure or homozygous. Pure or homozygous, that's what it's called. If it's big B, small b, then it is going to be still black, and this is a hybrid, and it's heterozygous, okay? Because they're different. And if it's small b, then it's going to be a white pig, but it's a recessive trait. Right? A recessive trait is always pure or homozygous because you need both of them to be present in order for the color to show up. Okay? So a hybrid black pig is big B, small b, and it's going to have babies with a white pig. Okay, so what's a white pig? A white pig is small b, small b. Okay. So we're going to do a Punnett square. So this pig, right, it's going to produce big B and small b gametes. Right? So half of the gametes, let's say this is the male pig, okay? I don't know what you call a male pig, a hawk or something, no idea. If it's the big B, half of the sperm would contain the big B and the other half would contain the small b. Okay, and then the mommy pig here is going to have small b or small b. Okay, there's no difference. So if these gametes come together, then you're going to get big B, small b, big B, small b, small b, small b, small b, small b. So 50% will be big B, small b. 50% will be small b, small b. Okay, so that means half of the pig should be black. The other should be white. Does that answer your question? Okay, so you can try the other two, right? Let's do number 4. The ability to curl your tongue on the side is it's dominant to not being able to curl your tongue. So this is big T for being able to roll your tongue, and not being able to do it is small t. Okay, so here we have a woman who can roll her tongue. Okay? So the woman is a tongue roller. Okay? If she can roll her tongue, then she must be small t, small t. Allison asked, would big B, small b be a mix of white and black? Not if it's a dominant and recessive situation. Okay, if it's dominant or recessive inheritance pattern, then it's either black or white. Okay, and in the hybrid condition, it will be black because that's the dominant trait. Okay, what you are asking, like when it's a mix of black and white, that's for codominance, right? That's when the heterozygous condition, both of the traits shows up. All right, so we'll talk more about that later. Hope that answers your question. Okay, so the woman here can roll her tongue, right? And the man cannot roll the tongue. Okay, so the man, the father, the man cannot roll the tongue. So what does it mean? That means he is going to be sorry, I got distracted here. Let's try again. The ability to curl your tongue is big T. So if you big T, or big T, small t, then that means you can roll. Roller. We say roller. Okay. If it's small t, then it is non-roller. Okay. So let's try again. The woman can roll her tongue. Okay. Woman is a roller. So if she is a roller, she's either big T, or big T, small t, right? We don't know. So big T something. And then she marries a man who cannot roll the tongue. Okay, so if you cannot roll the tongue, then it's small t, small t. Okay, cannot roll the tongue. And then what happens is they have their kid. Okay, they're gonna have a kid. So this is the woman, this is the man. They're gonna have the kid. Okay, and the kid, I don't know if it's a boy or girl. Let's just say it's a girl. Okay, so and the kid, has his father. So that's a square. Has his father's phenotype. Okay, so the child is going to be small t, just like his father, right? Okay. So what does that mean? Well, one of the small t came from the father for sure, and then the other small t must have came from the mother, right? So for that reason, right, we now know for sure that the mother will be big T, small t, okay, rather than big T, big T. Okay, so that's the answer for part A. Part B, what is the probability that their second child won't be able to roll their tongue? All right, so again, we can do a Punnett square. The mother is big T, small t, so half of her eggs is going to carry the big T, the other half will carry the small t. The man is small t, so, all the sperm will be small t. And if we do the Punnett square, we'll see that there is a 50% chance that the child will be small t, which is a non-roller. Okay, so that is the answer. Okay. All right. Let's do one more. Let's do one more. In humans, brown hair is dominant over blonde hair. Okay, so let's write down our possible situation. If you are big B, or big B, small b, that's going to be brown hair. All right, and it's going to be dominant over blonde hair. So blonde would be small b, small b. Okay, so if a woman with brown hair, whose father has blonde hair, marries a man with blonde hair. So that is like a lot of information all jammed up in one sentence. Maybe we'll do a little pedigree here. Okay, so this woman with brown hair. Okay, so brown hair is either big B, or big B, small b. So we'll put a big B, and we don't know what the other one is yet. Okay. However, this woman with the brown hair has a father who has blonde hair. So we go up one generation. Her father has blonde hair, so small b, which tells us that she must be big B, small b. Because her father is small b, then it's guaranteed he's going to be passing that on to the woman, right? So then the woman will marry the man with the blonde hair. So a blonde guy here is just small b. So what's going to be happening with their kids? Okay, what's going to be happening? Well, we can do the Punnett square again. All right, for the mother, it is big B, small b. So like this. And then for the father, small b. And if we just combine the letters together, you will see that there is a 50% chance that the kid will have brown hair. And then there's a 50% chance that the kid is going to be having blonde hair. Okay. So sometimes if you don't know whether, I mean, if the person has a recessive trait and you know for sure that genetic makeup is like, small p, but if they have a dominant trait, then you don't know whether they're homozygous or heterozygous. Then you must gain more information, right, by looking at either their parents or their children. Okay, that's it for monohybrid, single trait. Those are the easy ones. Okay, the dihybrid. Okay, that's the one that is going to be a little bit weird. Okay, we're going to try like number 4 right here. It's probably very confusing, but let's try. Hey, in rabbits, black fur is dominant over brown fur. Okay, so we have to write out all these things as soon as we see it. Okay, otherwise it gets confusing. If it's not confusing for you, good for you. It's confusing for me. So we write the big B. Big B is black fur. Okay, and that means small b is going to be brown fur. Okay. And then we can write out the different combinations. Okay, big B is black. So is big B, small b. Brown fur is small b. Okay, so that's the first trait. This is question number 4, right? Question number 4 from the second part. Then we have a second trait. Normal length fur is determined by a dominant gene. So normal length is going to be dominant over short fur. Okay, so we will say big N for normal fur, and then small n for short fur. So if the rabbit is big N, or big N, small n, then it's going to have normal fur. If it's small n, is going to have short fur. Now we have to do this part. Diagram a cross. A cross just means like to have babies. A cross between oh my goodness, homozygous black with homozygous normal length. Okay, with what? With a brown Rex rabbit. Brown Rex rabbit. Okay, so let's start with this guy. Homozygous black. So that's big B. Homozygous means the same, right? With heterozygous normal length. So that would be, homozygous normal length. That would be big N. Okay. Cross with a brown, so small b, Rex rabbit. Rex just means short fur, so small n. Okay. So determine the ratio of the F1 and F2 generations. So let's do F1 generations. Let's write out all the gametes. Okay. Now, if the rabbit number 1, it's big B, and big N. Right? So we are looking at a chromosome like this, right? Chromosome, and we have big B here and big N here. We actually don't know if they're on the same chromosomes, but so let's maybe not do that. But 100% of the gametes would be big B and big N. Right? There's no other combination, right? You guarantee to get a big B, and you guarantee you're gonna get a big N. Right? Same thing for this one. Guarantee big N, guarantee small n. Okay, so if we do the first generation, the F1 generation, okay, we put the big B, big N gamete here from one rabbit, and then we put the small b, small n from the other rabbit. Okay, so that gamete goes there, that gametes go here. Then what do we have? Okay, in the F1 generation, 100% of the bunnies will be big B, big N, small n, which means they will have black normal fur. That's the easy part. Are we okay? Now the question asks us to do F2 generations, right? Okay, so what does that mean? That means we're going to take 2 rabbits from the F1 and we're going to cross them. All right, we're going to take 2 rabbits from the first generation and we're going to cross them and see what happens. Okay, so over here, let's write out the gametes. Okay. So what are the gametes going to be? Well, this is kind of like math. Okay, it's like when you do your polynomial expansion. Okay? You're going to take the big B. Big B can combine with big N. And big B can combine with small n. We can have small b with big N and small b with small n. Okay, so the possible gametes are big B, big N, big B, small n, small b, big N, or small b, small n. Okay? This is the exact same genotype as the other rabbits, so you're going to have the exact same gametes. I hope you're doing this with me, right? Not just watching. Okay. So we have 4 possible gametes here with another 4 possible gametes. What are we going to do? Well, we're going to have a big chart, very big chart. Okay. Let's put the gametes here. Big B, big N, big B, small n, small b, small n, a big N. Like that. Okay, so these are gametes from one parent. This is gametes from the other parent. Okay, again, I'm going to give you a moment to catch up. Now we're going to combine the chart. Okay. So you should be able to do it yourself as well, right? You take this and this together, you get big B, big B, big N, like that. Okay. So big B, big N, small n, so on and so forth. All right. Let's try the second row, second column. Big B, big N, small n. Okay. You should double check my work. Make sure I'm doing it right. Okay, so those are all the possible combinations. All right. Let's try to find out everything that looks the same. Okay, so over here, and I need your help for this, okay? This is big B, big N, big N. What will this rabbit look like? For a rabbit to have big B, big N, what does it look like? Let me out here. Okay, yeah, a few people said they're going to be black and normal fur. That's correct. Okay, so I want to find out if there are other rabbits that will be normal, black and normal fur. Black, normal fur. Okay, so that one is black and normal. And I want you to highlight everything that's also black and normal. For example, this one is also black and normal fur, right? Okay, so please try to identify all of them. I'll give you a moment to do that. Okay, if you have, you can type them in the chat box if you want, or you can just highlight it on your page, I guess. Okay. Yeah, thank you. And if you do all of it, it will look like this, right? Okay, I think those are all of it. That would be black, normal fur, normal black fur. Right? And if you count all of it, it will be 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 will be black, normal fur. Okay. Let's try to identify another type. Okay, so there's another guy here. This is a black and short fur, right? Black and short fur right here, and then other black and short fur. Okay, so 3 out of 16 will be black and short fur. Okay, these are the green ones. These are the yellow ones, so to speak. Okay. We will have these guys right here. What would they look like? Are they brown or black, short or long? What's going on? Three out of 16. Those would be brown and normal length, right? Normal fur. And last but not least, there is a small chance, 1 out of 16, that it is going to be brown and short fur. Okay, so this is the classic 9-3 to 3-1 ratio. If you start with purebred in the first, in the original parents, okay? If the original parents are like pure on both traits, in the second generation, F2, you're gonna have the 9-9-1 ratio. Okay. I'm gonna show you how I'm gonna test you on the test. Okay, so you might get a question like this. I'll let you read it first. Okay. And I'll tell you how to do it. As you can see, right? You can probably tell based on the format of the question here that it's a screenshot directly from your quiz bank, right? Okay, so I wouldn't be surprised if you get one of these. All right, so I assume you've read it. If you haven't, I'm gonna read it now anyways. So true breed just means like purebred, right? Purebred. Okay. Pure breed means a pure breed, which means they are homozygous on all the traits, right? Just like the rabbit question, right? The rabbit question right here. Where's the rabbit question? Okay. The parents are homozygous black, homozygous normal. So that's a purebred or true breed. Okay. I don't know if you guys like dogs. I personally don't like dogs, but I know people who like dogs. Sometimes they want to buy like pure breeds, right? Okay. And what does that mean? Well, it just means that they are homozygous on all the locus. So if you take a pure breed and then you breed it with another pure breed, then all their kids, all the puppies that comes out will preserve those traits, guaranteed. That's what pure breeds means. It doesn't mean is going to be a good looking puppy, although people usually would, like to have pure breeds for traits that are desirable. But you could also have, like a subjectively ugly a dog that is also pure breed, right? If all the undesirable traits are homozygotes. Okay, so pure breed doesn't necessarily means like, you know, better looking, good looking. It just means it's homozygous on all genetic loci. Where was I? Right here. Okay, so 2 pure breeds of peat plants across. So one parent has red and axial flower, and then the other one has white and terminal. Okay, so here it says all F1 generation, all F1 individuals have red axial flower. So that's important information because the parents are pure breed. And if we look at the F1 generation, the traits that shows up in the F1 generation will tell you which one is dominant. Again, we go back to the rabbit question that we did earlier. Where is it? Right here. Okay, you see, the parent here is black and normal length. This is brown and short length, and then all the F1 generation is black and normal. Okay, these are the same trait as the parent with the dominant trait. Okay, so you can always tell by looking at the F1 generation. If the parents that you begin with are pure breed, then the F1 generation will exhibit the dominant trait. So based on our question here, it says the F1 individuals have red and axial flower. That means red is dominant over white. Okay, and axial is dominant over terminal. Okay, terminal means the flower is at the end. Axial means is like in the middle of a stem, I suppose. Okay, so what are we gonna do? We are gonna have 1,000 F2 offspring. Approximately how many of them would you expect to have red and terminal flowers? Okay, so some of you might be thinking right now, yo, this is not fair. This question is so involved and it's like just 1 mark, right, on the test. And I have to do so much work and waste so much time, you know, doing this. It doesn't make sense. Well, I kind of agree with you. However, you actually don't have to do a lot of work, okay? What you have to do is this. You have to realize, oh, this question is just like the bunny questions, okay? So you're going to take this chart. And you're going to stick it on your wall, okay? You're going to stick it on your wall. Hold on a second. I just have to copy this. Copy and paste. I'm going to put it right here. There we go. Oh my goodness, what is going on? It did not come over. Just give me a second. Let me try it one more time. Sorry, having a not so smooth day with these things. Try one more time here. Yeah, Tyler's already giving me the question. Yeah, at the answer. That's good. Over here, right? That is the chart that you need. Okay. And we are looking for the number of offspring that is red and terminal. Okay, so red is the dominant trait, and terminal is the recessive trait, right? Okay, so if you remember, dominant means the capital letter, and the recessive means the lowercase, right? Okay, so which of these is dominant combined with a recessive? Well, it's all the green ones, right? All the green ones. So that's 3 out of 16. Okay, 3 out of 16. Multiply by 1,000. Right? So what does that give you? I have no idea. Let's take out our calculator. We will have 3,000 divided by 16, which is 187.5. Okay, so the closest answer is going to be, what is the closest answer? 187.5. So I'm going to say E right there. Okay, that's what it means. So you don't have to do it again. Right? Now, what if the question says, how many of them would be white and terminal? White and terminal. Right? Which one of these ratio is white and terminal? Well, both of them are recessive. So white and terminal is this one, which is 1 out of 16. So you would take 1 out of 16 times 1,000. Right? So what's 1 out of 16 times 1,000? That would be like 62.5. So the closest answer would be 65. Okay. It's not going to be exact, right? Like just like statistics, right? If you flip a coin 10 times, statistically, you're supposed to get 5 heads and 5 tails, but we're not going to do that. We're not necessarily going to get 5 and 5, right? So same thing. Statistically, you're supposed to get 187 and a half being red and terminal, but 190 would be the closest one. Does this make sense? Like if you see this question on the test, you understand. You can just find this chart and then just look it up, right? So that's why it's 1 mark question. You can actually do it very quickly. Okay. The key is to have this chart though. Okay. Stick it on the wall. I was actually looking at the results of the previous quiz number 3, and there are some questions where I give you the template strand and I ask you for the RNA, right? And then there are some questions where I give you the coding strand and I ask for the RNA, right? So a lot of people still get mixed up, right? But last class I gave you this assist mode, right? This cheating sheet thing, right? So I don't know if you guys were using it, right? But if you see that, if I give you the template and ask you for the RNA, you can see that they're complementary, right? And then the coding strand will be identical, but the U replaces the T, right? So whenever I tell you to, put this thing on your help sheet, you should take advantage of that. Okay? It's going to help you. All right. Let's do another question right here. In a cross of this, what is the probability of producing a genotype of, big A, big A, big B, big B, and big C, big C? All right, let's take a look at this. For this particular question, okay, let's take a look at big A, small a, big A, small a. So if I have big A, small a, sorry. If I have big A, small a, and then big A, small a here, right? And then I do the Punnett square. Okay, like that. What's the probability of producing big A, big A? Well, it is going to be 1/4, right? 1/4. What about big B, big B? Well, it's the same scenario. So if I do another Punnett square, it will also be 1/4, right? So I can times another 1/4. And then same thing for the big C times another 1/4. So 1/4 times 1/4 times 1/4 is 1/64. So that would be the answer. Okay. Do we have another one here? Here is another one. How many unique gametes would be produced through independent assortment for these alleles? Okay, so I'm gonna launch the poll. Oh, sure. Let me go through that again. Okay, so first of all, the probability of multiple things happening at the same time is just the product, the multiplication of the probability of each individual event happening. For example, let's say what's the probability of getting a head on flipping a coin plus rolling an even number on a dice? Okay, so the head is 1/2, right? Okay, and rolling an even number, right, as in like 2, 4, 6 on a dice is half. So the probability of both of these things happening together is just you multiply the individual probability together. Okay, so whenever you want the overall probability, you just multiply the individual probabilities together. Now, understanding that, we will take a look at this again. If you have big A, small a, big A, small a, here is the Punnett square for it, and you can see there is a quarter chance that you will get big A, big A. Okay, so that's the first quarter. Now I can do it for the B as well, right? Big B, small b, and then big B, small b. So if I do the Punnett square here, you can see there is also a quarter chance of getting big B, small b, right? Big B rather. Okay, and then you can do it again for C. And again, you have a quarter chance, right? So a quarter times a quarter times a quarter, that's 1 over 60, 64. Okay, what about this one? Okay, we are kind of running out of time, so I will just do it, and you can see if you understand. How many unique gametes could potentially be formed? Okay, so let's take a look at, let's write a little bit bigger. A, B, B, big C, big D, small d, big E, big E. Okay, so how many possible gametes from big A, small a? Well, you can either pass on the big A or the small a, so that's 2 possibilities. Same thing for this one, either big B or small b. This one is only 1 possibility, okay, because you only have big C. 2 possibilities here, 1 possibility. So if you multiply all of them together, 2 times 2 times 1 times 2 times 1, that's 8. Okay. Let's do one more. So let me see if I can make this bigger. So basically here we have a pedigree, and there's a dominant trait, which is like something called woolly hair. So big W is woolly, okay? And I suppose small w is normal hair. What is the likelihood, the probability that the children of 4, 3 and 4, 4 will have woolly hair? So 4, 3 is right here. 4, 4 is right here. We just look at the Roman numeral, tells you which generation we're looking at, and the number tells you which individual in that generation. Okay, so we want to know the probability of this couple having a child with woolly hair. Okay, so let's see. This person, number 4, does not have the woolly hair because it's not shaded in. So we know this is small w, small w. Individual number 3 has the woolly hair, so at least there must be 1 big W. But what about the other one? Well, we have to trace back to his parents, and we can see his mother has normal hair. So normal hair is small w, which means he is guaranteed to have 1 small w. Okay, the big W came from the father, right? So now we do the Punnett square. Big W from this individual, small w from this individual. And when we do the Punnett square, we see there's a 50% chance that the kid will have the woolly hair. The answer is 50%. All right. Okay, let's move on. I think I'm gonna skip the incomplete dominance and codominance for the moment because we don't have a lot of time. I mean we can review it again, right? Like later on, but let's talk about the sex link. The sex link is usually what people get confused about. Okay, so this is the sex-linked problem. So when something is sex-linked, it means that the trait is passed on the sex chromosome as opposed to the autosomal chromosomes. Okay, so there could be X-linked, which means it's passed on the X chromosome, or Y-linked. If something is Y-linked, then only males can get it. So here in fruit flies, one white eye color allele is sex-linked. Okay, so XW is the white eye allele, and then the normal is a red eye, which is dominant over the white eye. Okay, so let's take a look at what this means. Okay, so let's see female. Okay, female. So female is XX, right? So there are three possibilities here for the fly. It could be XX, which means this is a red eye female. Okay, it could be XXW, right? So the female fly has one white eye allele, but because the red eye is dominant over the white eye, this is still going to be red eye. Okay, female. But because she carries one of the white eye allele, we call her a carrier. And then if it's like this, then that would be a white eye female. Okay. For male, it's going to be XY. So this is a red eye male. And then because male only has one X chromosome, so if the X chromosome is the white eye allele, it will automatically be white eye. Okay, for female, they would need both copies of the white eye in order to show up. Okay, so these are all the possible genotypes and phenotypes of an X-linked situation. Okay, so question A. We have a white eye female crossing with a red eye male. Okay, so white eye female, as you can see, is this one, right? So XW cross with a white with what? Sorry, with a red eye male. Red eye male is right here. XY. Okay. So XW, we're gonna put it right here. And then X and Y is just gonna be like this. So if this sperm fertilizes this egg, we will have XXW. Same thing over here. XWY, XWY. Okay. So that means there's going to be a 50% chance that the child, the offspring, we should say, the offspring is going to be a red eye female. Okay. And then there's going to be a 50% chance it is going to be a white eye male. Okay. So that means if they're going to have a female offspring, all the female will be red eye and all the males would be white eye. Is there any question about this? Okay, we'll try another one. Both Frank and Kate have normal vision. Their son is tested positive for color blindness. What is the probability that their daughter is colorblind? So color blindness is an X-linked condition. You should know that. So XX. Could you go up for a second? Okay, sure. Sorry. Let me know when you get. Okay. So both Frank and Kate have normal vision. Their son is tested positive for color blindness. Okay, so let's do this. There is Frank. This is Kate. Their son is colorblind. Their daughter, we don't know. Okay, so the son is colorblind, which means the son is XCY, right? XY for male. If it's colorblind, then it's the colorblind allele on the X chromosome. Frank and Kate both have normal vision. So what's the genotype for Frank? Well, it's just going to be XY, right? Normal X. Right? And Kate, can someone tell me what is the genotype for Kate? There are three choices. Is she XX, XCXC, or XXC? Which one is it? She has normal vision. Well, we know she is going to have a normal X for sure, but what is the other one? Well, let's take a look at their son. The Y came from Frank for sure, because the male is the one that passes on the Y, right? Always. That means this XC must have came from Kate. Okay, so she is a carrier of the color blindness gene, but she herself is not colorblind. Okay. So the question is, what's going to happen to the daughter? Is their daughter going to be colorblind or not? So let's take a look. Frank here is going to pass on either an X or Y. And for Kate, it's X and XC. So if I do all these, right, we'll see. Excuse me. We'll see that these are the genotype for the daughter, right? So the daughter here is either XX or XXC. We do not know for certain. Okay, but what we do know for certain is whether she's XX or XXC, either cases, both of them would be normal vision. Normal vision. She might be a carrier, but doesn't matter. She will still have normal vision. So the question asks, what's the probability that their daughter will be colorblind? No chance. 100% guarantee. Make sense? That's actually the situation with my uncles and aunt's family. Their son is colorblind, and then they want to get their daughter tested. I told them, you don't have to test her. She's guaranteed to have normal vision. But nobody listened to me. Anyway, she doesn't have colorblind as predicted. So that's that. All right. Next week, we don't have tutorial. We will have, you're supposed to do your lab instead, okay? So I'm going to spend some time and talk about the lab right now. However, okay, I know this stuff can be a little bit confusing. So if you want to have like a Q&A session next week during this time with me, why don't you send me an email, okay? So if enough people, or like even if it's just 1 or 2 people, okay, who need extra help next week, I will still come to the tutorial, okay? So there is no official tutorial next week, okay? However, if you need extra help, either with lecture 8 or even with these codon stuff, send me an email, and then I will still come for the tutorial. Does that make sense? Okay. If you have no question, then like don't come. Okay. Like if you have no question, you show up, we'll just be staring at each other. But if you do have questions, send me an email and say I will come, and then I will host it. Okay. Now let's talk about the lab. Give me a second. Okay, so for the lab, some of you already handed it in, which is great. I'll provide a link to the lab in today's debrief, but basically you can find it under lab 2. So you should watch the video on how to use and care for a microscope. And then there is a link for the virtual microscope. Okay, I want you to open that. Okay, you might have to click it twice. I'm not sure if it's gonna run on your phone. It's definitely better on your computer. Okay, so you actually have to take a lot of screenshots for this lab. Okay, and this is loading, and the actual lab is right here, microscopy lab. Okay, so I will click on that. All right, and it tells you exactly what to do step by step. Okay, don't just skip all of it and then just go on to the questions. Right? Because there's a lot of instructions. I highlight all the questions for you. Okay, so you know what to look for. But in order to answer the questions, you have to read all these things, right? Okay, so it tells you to click on main to return to main menu, click on explore, and that's what you're going to do, right? You see all the button here, right? Okay, so click on explore, for example, and then you can open the case. I know this is really kind of lame because it's like virtual, but it's the best we could do at the moment. Okay, so you might have to fine focus it, and get a sharp image. And then I'm gonna ask you to take a picture at 4 times, 10 times, 40 times. All right. So this is what you should not be doing. Okay. Do not take a picture like this. Actually, I have to share my whole screen for you to see what I'm doing. Screen 1. Okay. Do not take a picture of the entire screen like this. Okay. Like you see what I just did here? Like some of you take a picture of the entire screen like that and then paste it into the assignment. Like, don't do that. Okay. I just need to have this particular view. Okay. And then you can paste that in. All right. And then you can make sure it's focused and all that, right? You can take it with your phone. You can take it with a screenshot. And if you use Mac computer, I think it's the little command key and then G or something. Okay, you can Google it if you don't know how to take screenshot. All right. Now later on, the question will ask you to find a cell that is in interface or something like that. Okay, so all you have to do is follow the instructions. Okay, but when you get to this screen, okay, again you have to focus it first. Okay, you obviously cannot see the cells very clearly, so you have to zoom in. Okay, you zoom in, and then I'll zoom in one more time. Okay, so I want you to find a cell that is in interface, right? Like that would be a good shot, right? Okay. What I don't want, and you will lose mark for this, is some people would leave it at 10 times and then just send me this picture. Okay, like I cannot really see clearly, right? So make sure that your picture is definitive, and there's no, no doubt about what is it that I'm looking at. Okay. What else is there to talk about for the lab? I think most of it is pretty self-explanatory. Let's take a look at this one. I'm going to give you a free answer because I get wrong answers from that question every time, and it's a little bit annoying. So I'm going to give you the right answer for this. Okay, so the question asks you, what is the relationship between the image that you're looking at? You're looking at the letter E. What is the relationship between this image and the original picture? Can you tell me? Anybody tell me the right answer, please? The relationship between this E and a regular looking E. What is the difference? Well, I mean, what's the relationship? It's okay. You can tell me what you think. Okay, and then if you get it wrong, I'll tell you, and then you'll have the right answer. But I want to hear what people say. This is the question, right? What is the relationship between the image, the E, right, and the object? Okay. Crystal say the E is inverted in the microscope. That is it. Okay, that is the right on. That's the answer. I'm looking for the word inverted. If you say this is upside down, that's not the answer. Okay. If you say it backwards, that's not the correct answer. Okay. It is both upside down and backwards. Okay. When it's both upside down and backwards, we say it is inverted. Okay, so make sure you write that down. Okay, it's upside down and backwards, which means it is inverted. To demonstrate the properties of inverted, Okay, besides the letter E, what other letter can we use? Okay, can we, for example, use the letter S to demonstrate this? If you look at the letter S under the microscope, is it going to be apparent that it is inverted? You know, S. I just typed the letter S, right? And I hope you can see that it is not going to be apparent because the S is symmetrical. When you rotate it 180 degrees, it looks exactly the same as the original one, right? So the letter S will not be an appropriate letter to demonstrate this property of the microscope. All right, so you will have to think about what letter to use. All right. Now there is also a couple other things before we. I know it's time already, but I have to talk about this. There is a YouTube link where I show you how to do a sample calculation. Okay, so the sample calculation is only a sample. Okay, many people copy exactly what's in the sample calculation and put it right here, and it's going to be wrong. Okay, because this is a different question. Okay, so the numbers, the calculations will be the same, but the numbers that you use will be different. Okay, so please pay attention to that. You can watch how to do the calculations, but when you actually sub in the numbers, make sure you're using the right numbers. Okay, based on this particular question. All right. I don't know. Sometimes the weird symbol shows up like this. Just ignore them. Okay, this is just DFOV1 times magnification 1. Don't worry about this upside down question mark. I don't know why it is there. The rest of it should be pretty self-explanatory. If you run into any confusion, feel free to email me to clarify. However, when you email me, this is very important. Because I'm not at my computer all the times, I am at my phone all the times though. Okay, so when you send me an email, don't just say I don't understand question 3 because I won't have question 3 with me. Okay, so type out what question 3 says, then I can get back to you quickly. Otherwise, you have to wait until I get back to the computer, open the lab, and then that will slow down how quickly you get a response. All right. So that is that. I mean, I was going to take up the quiz, but I guess that can wait until later on. And that's it for this week. Okay, so again, no official tutorial next week. However, if you want to see me for extra help, if you have specific question, send me an email, and then we will have a session. If I receive no email, then there will be no tutorial. I guess I'll post an announcement, okay, to see whether I'm hosting one or not. Okay, but email me if you need help. Okay, thanks a lot for coming, and I will see you all, in 2 weeks if there is no extra help. Bye for now.